Tribeca had a series of portfolio companies, one of which was Spot On Sciences, which had these innovative dry blood spot technologies. And so when I was first brought on at Tribeca, that was the company that I primarily focused on. And we had a pretty innovative, good plan. We entered into the concept of the microsampling world and how do you really take these dry blood spot technologies, which are key enablers in order to expand kind of the utility and the reach of clinical testing. How do you take these technologies and grow them in the industry and commercialize them in such a way that you can get adoption at a broad use level? And that led us to the fact that we needed to have a laboratory, which was critically important because getting someone to transition from blood or serum onto a dried blood spot technology is a challenge. They have things that work for them already. There's a huge cost. There's a huge level of effort. So we needed to partner with a laboratory that could help us do the adoption and kind of drive the protocols behind some of these key areas in which we wanted to grow in terms of the market sectors we were most interested in playing in. All of that was very exciting. We had a whole plan. We had our marching orders, and then kablammy, COVID hit. And so it was time to restructure. It was time to rethink. But the two things that we knew is we had a core capability of being able to do at-home sampling, which we thought was very important in this pandemic situation. And we had a laboratory that had all the capability to ramp up into COVID testing. It was traditionally a genetics and PCR laboratory anyway. So it was a perfect marriage to say, let's pool all, the bright minds and the great people that we have here that are working in these diverse portfolio companies that hardly have ever been met. Let's get them all together in a room and let's solve this COVID problem as best we can. And the solution we came up with is threefold actually. First, we got our laboratory partner to come on board with us and get all the EUAs and dot all their I's and cross all their T's so that they could meet all the requirements for COVID testing in a CLIA certified high complexity lab. We're doing both PCR testing and antibody testing. And then the second thing was we had these beautiful home sampling devices. So how do we get to the point in which we can transition out of traditional sampling in a clinic or at a point of care and get to in-home sampling? And that's something that we're working on doing today. We're not there yet. Obviously, we are at the beholden of the FDA and the rules and policies in which they want to see things roll out, but we're definitely working with them closely. They've been very helpful. They're informative. They definitely keep us in the loop on what we need to be doing in order to achieve that. And then the final thing is when we consider the COVID testing space and what it means to bring this into a workforce, it's a very different scenario from the typical show up at Mass General, get your test, walk away, goes back to your physician and you get a result and then they work with you on how to treat you. So at home or workforce testing has a very different model. It kind of cuts out some of the key people like the physicians that would necessarily be involved in the aftercare, but this is more to just get some baseline screening and some understanding of what is the COVID status of your market. So before I get too far into that, since I'm not advancing the slides, let's go in here. So you can see here, this is our collection kits are to the right of the screen. And what you see to the left is the HemaSpot SE device. This is our serum separating device that we use in our antibody testing kits. Right now, currently, we don't do anything in the home. We do it all under healthcare supervision. But you prick your finger, you apply the blood to the device. The blood sample actually separates out from the whole cellular components into serum. So you can see the red spiral begin to kind of turn yellow and kind of wrap around the loop. Well, that yellow portion is actually the serum. You close it up. There's desiccant on board. It will dry the sample in just a few minutes, and then it's ready to be put in the mail and shipped for analysis. So the labs that are using this are doing the antibody testing, and we have both an IgG and an IgM antibody test that have been validated using our devices. So that's exciting. And so then the other test is,
Questions are already pouring in. May I take you off script for just a second and ask them straight away? First, is the HemaSpot SE authorized through the emergency use authorization? Is this a product that we can get today, yes or no?
Well, so it's been validated in the laboratory with the assays, and so without doing it at home, yes, it can be used to collect samples with healthcare worker supervision present, and it can be shipped to the lab and run on the assays. It doesn't need itself an EUA, right?
It doesn't need
I hope that I'm saying. I might be saying that wrong. Maybe Jana, she's probably better on the regulatory side. But from my understanding,
I just emailed her again to see if we could catch her. Let me see if she's among the attendees
yeah.
here. Oh, yeah.
But if we were to do it,
hold on a
yeah,
second.
can we turn her on?
Yeah, that's what I'm going to do. Let's see. Okay, Dana needs to, and I don't understand this pin business.
Maybe I'll have Dana, she can dial my phone and I'll just put it on speaker.
Yeah, I think so. She can.
Dana, can you call me?
And until she jumps in, we don't have a regulatory, but all of these do require EUA authorizations, and it's different for each setting. So, if it's in the workplace, that would be one. If with supervision, if it's at home with supervision, that's another EUA, and if it's without supervision, also that's another authorization.
Jeremy,
so right now what we're able to do.
I was a bit distracted with. Oh, it looks like Dana's phone just became green. Dana?
Hi, good morning. Sorry, I've been listening and sorry about that.
It's okay. Do you have a webcam?
Yes, and I will work on that as you are continuing to talk.
Okay, Jeremy, hooray. Now,
and
Dana, what's your role?
Morning.
As soon as a regulatory thing came up, Melissa
Dana's
was like,
our chief medical officer, so everyone knows, and is very knowledgeable in the regulatory side of things. I'm more the clinical side and implementation.
So my question was, and really the audience is, I think Jeremy began to address it, and I got a little distracted with the technical stuff. Do you have an EUA, or do you have multiple EUAs?
So that's a great question, Joe. We are in the process of submitting multiple EUAs. So right now we have the EUA for on-site testing with a healthcare provider doing the sampling. We're in the process of submitting our EUAs for a home supervised version of the kit with a healthcare provider actually watching the sampling, which we found to be very important for the B2C model, B2B model, because employers want to make sure that the person that's sampling, that we're seeing them. It also helps, promote some goodwill as we're going through the sampling. And we also are in the process of submitting an EUA for a home kit collection without a healthcare provider on site.
Okay, thank you for that. Second is,
Sure.
As I know you're amply aware, FDA was, I won't say lax, freewheeling in letting tests flood the market, and it was just, it works, right? Sure does. Okay, great.
Yeah.
Just on 60 Minutes, perhaps you saw it this weekend, about a town in Laredo, Texas that is out $500,000 having bought basically nothing.
Oh, yeah.
Yeah. So I'm unclear, given all the products that have already hit the market, how do we, what's the company's feeling on how do we, I won't say restore, establish confidence that any test really works? All I keep hearing is false positive, this and that. So how do I? Anyway.
I can answer that, but I also believe Melissa, as our scientific expert, can help with some of that. I can tell you, we've felt the same thing. When we initially started this project, we called it the Wild Wild West. Everybody was just rushing to do something, trying to help out. We've been very precise in how we've addressed FDA, because it is important, and Melissa can talk about the sensitivity and specificity. PCR testing is essentially pretty much the same, but as I think your audience was hearing prior to my actually figuring out the audio and the visual, our HemaSpot SE is our pride and joy, and we want to make sure that when we do bring it out, that we've crossed all the T's, dotted all our I's, so that it does not fall into this category of inappropriate testing, because this is actually useful for things. I don't know if Melissa we got far into the HemaSpot technology, but we're using it in South Africa for AIDS, for example, and it has a lot of potential to sample other devices, other diseases. So we absolutely do not want to misstep with FDA on this.
Let me understand. Did you say that this product is on the market with other indications, or you're just bringing it to market now for this, but has other indications in the future?
Currently, the SE device, all of our devices in the Spot portfolio are labeled for research use only. We are in the process of getting a 510K clearance for these devices, and we're going to do a Class 2 clearance, so it doesn't necessarily have to be one indication, one clearance. We want to get a broader range of clearance. So we've been working with the FDA over a year on that, and that's well in progress. It's just that this whole pivot into COVID has actually given us an opportunity through the EUA in a real crisis situation to flip this switch on and to be able to start using it for diagnostic tests, I guess for antibody testing today.
And you said 510K, so that indicates there's a predicate. What's the predicate?
That has been our biggest hurdle because we've hired a very expert regulatory group to help us figure out what the predicate is, because the device itself is so unique, because it's not, and again, Melissa is our scientific expert here, but this isn't regular dried blood paper. There's actually a glass filament within the paper that creates the separation. So finding a true predicate has been a little bit of a challenge for us, and we think we have a really good path forward and are submitting that actually, hopefully very, very soon.
Okay. I have another question here. This one's from Alan Connor. How long does it take to get results from the Hema Spot?
Well, it takes about 5 minutes for the sample to dry before you can ship it. And then once you ship it to the laboratory, it'd be like anything else. It requires an extraction process. So basically, for a couple hours, you put it in a buffer. You take a cutting or like a 6 millimeter punch, 1 or 2 punches. You put it into a simple PBS buffer. You elute off the serum components that are left on the paper, and then you'd be ready to go into your PCR assay.
Okay, and Alan, if you have a follow-up, you're welcome to ask. Bill asks, how about IgA? It's a respiratory virus. Any plans?
Not at this point. We have no IgA assays that are in our rearview mirror that we're considering bringing on board. Now, we watch the market very carefully. We're always trying to keep abreast of where the transitions are and where we're going to get the most informative information to make the best use of the technology that's coming out. So it's certainly not off the plate, but there's nothing that we're pursuing in that vein right now. If you have IgA assays, we're happy to hear about them.
Okay, and Jeremy, I think, is in the chat room, so we'll be able to keep in close touch that way. And of course, Shannon is, so we can talk that way as well if there is that kind of development. Why don't I let Jeremy, God bless you. Your house is a rocket. I'm going to put you back on mute, and you can sign language when it's time to be heard, okay? Are they going away? Your lovely, lovely family. In any other situation, I'd love to play with them, but right now we can't hear ourselves. Yes?
Okay. It sounds like I don't hear it anymore. I think you muted it.
Silence himself. Okay. Why don't I let you take us to the next slide, and I'm sure the question.
Well, that's really quick. I just wanted to highlight too. We have 3 kits here in terms of our COVID product offering, and one is for antibody testing. Like I said, we test both IgG and IgM, and then we also have the diagnostic kit, and then we do a bundled where you can get a combination of antibody and the diagnostic assay together. And if I go to the next slide, which is not advancing. There we go. And I just briefly want to go over this because I'm really far more interested in hearing from the people who are on the call, their feedback and understanding of where things are going with COVID testing in their own workforce, because I just feel like this is a great opportunity to put our heads together. I think there's a lot of really very knowledgeable people, very connected people, a lot of decision makers. So I'd love to just get a sense from the audience of how they're dealing with this and what their thoughts are and what they think the value of testing really is in terms of bringing your workforce back online. But with that, I'll run through this briefly and then we can get on to the good meaty discussion. So again, the way in which we positioned our program is we really wanted to not just do like a test one and done. We really wanted to, especially in the workforce environment, offer a service that would give the employer some visibility into what was happening in their employee population, and then, provide on top of that consultative services going forward, so that decision making was informed and they had someone to lean back on to have some sensibilities on what good decisions might be or what best practices might be. And from the discussions we've had, it's been really fascinating. Most of our discussions with customers who we're bringing on board, it is much more of the time is spent educating, not even talking about pricing or the program. It's just, we can get into, an hour and a half discussions on just educating them about what they're hearing on the media and what this means, making clear lines between what a rapid test is and what a laboratory test is. And it's been an amazing experience in that it's such an untraditional sales opportunity space. And really, I think that it's easier for people who don't have to go into work to just put off bringing people back and not test, because it's just such a complicated quagmire of disinformation, conflicting information, and uncertainty. And so that's what we've tried to do with our program is highlight this, say that we understand it, let us help you understand it, and make smart decisions that make you feel confident, and also enable your employees to have confidence that they can come back to the workplace and it will be a safe environment because you're taking the precautions. And it's not 100% testing by any means, but all the precautions that can be taken are in a rational, thoughtful, and informed way. So so I'll walk us through this slide very quickly, and then I think just maybe one more after this, and two more after this, and we can get into the heart of the dialogue. But the way in which we've set this up is we have a portal. Employers will sign up they'll send an email around to their employees, and then the employees will sign up and make appointments for testing. And they can do all the scheduling, and they'll do the screening questionnaire for eligibility. It will all be done in an automated way. And then from there, they will come on site, and they'll show up on site, and we'll go through the schedules and the appointments and do the collection. After that, all the samples are sent to the lab. We have CLIA certified high complexity laboratories that run the tests and deliver the results. We have a HIPAA compliant portal in which all results are returned to the employees, and then in a de-identified way, the results are also given to the employer. We also have a software platform that will then aggregate all this de-identified information for the employer and let them take a look at kind of the overall status of their workforce. And then afterwards, as questions arise or as people, sorry?
Nope.
Oh, okay, yeah. As questions arise, or as, say for instance, someone may come down with COVID, we can turn around a rapid test and, we can turn around a testing opportunity to get them in and get them swabbed, and then, then we can get the results. We can identify that they are positive for COVID. We can also then help determine when the right time for them to come back to work might be, considering the kind of clinical indications of their infection and how they're cycling through it. So again, we didn't want to be a one and done testing organization. We really wanted to be a partner to an organization that has to deal with this over a long period of time. COVID is very dynamic. It continues to move. It ebbs and flows, and you see spikes and valleys. And so we knew that going forward, it was not going to be simple. And that, again, this kind of one and done is, sorry?
I'd like to see some valleys.
Yeah, we'll get there. Yeah, but yeah, and again, different states in different places are all in a different position. And so it is a very dynamic, complex environment. And so as best we could, we wanted to bring the brightest clinicians and physicians and people on board that could really be of value in helping to navigate this. Because, I'm not going to say we're in this forever, but, we've got a good extended period of time, 18 months, perhaps, maybe less if we're lucky. But it's enough time that the world needs to get back and become operational. The economy needs to return. Or, I don't know what the future will hold if we can't bring people back to work and we continue to have these massive amounts of money to continue to keep people financially afloat while nobody's being brought back to work. So, and again, this isn't well suited for everyone. Some people, it is totally feasible to work from home, and it's not an issue, and it's probably a much safer, easier, kind of less encumbered choice. Whereas other organizations have to go back. They have essential workers. They have things they manufacture. Our lifestyle and our ability to have core essentials to feed our family, and we need those people. So how can we support them? And that's how this all came about. And I'm just going to, here's an example of what the test results look like. You'll get a positive or a negative, or if you happen to get an inconclusive, we have to retest, we'll let you know. But we do have physicians that when a positive result comes back, we don't just walk away and say, here's a positive. We have a physicians network that will follow up with the patient, provide advice, encourage them to connect back into their primary care physician and continue the treatment through, the right channels so that they get care all the way along through the way of their
These physicians are employed by whom?
We have an organization that we've contracted with, which is a team of physicians that do a type of telemedicine business. And so we've contracted with them, and they're going to serve as the physician, kind of people who are guiding the process, looking at the tests, issuing test orders, and following up with the patient should they have a positive outcome, or should they have a negative outcome, but with clinical
patient.
Sorry?
Directly with the patient, not the employer?
They interact with the patients, correct.
And the employer, though, right? You do say that Joe Hage is sick, right? Well, everyone says that, but you get the idea.
Yeah,
I would
Joe, if
prefer
I
to
may.
Dana Welling.
Please.
Through the online portal, the employee has to consent to have the services done as an individual, understand the risks and the benefits of the test. They also have to give, because of the HIPAA privacy laws, specific permissions to have the employer view only the results of the test. So as you know, likely know, with right now, because it's a pandemic, the EEOC and the ADA have said that employers may mandate screening and testing, PCR testing. And because of that, we've been able to tell employers that they are able to offer this to their workforce writ large. And if a positive result comes back, the employer will get the results back. We as a company will be responding to the patient directly and speak with the patient about what they need to do moving forward to make sure they're healthy. Mostly it's around referring them, making sure they connect with their local primary care provider, letting them know of their test results.
I'm just now, as you say it, thinking about, for the first time, I guess, the incremental economic burden, I presume, to the employer to do these tests. They're paying you, right? The employer pays you. And I'm thinking it's not inexpensive. So here they are already depressed with their ability to earn revenue, and here's yet another expense to try to get operational again. Thank you, COVID.
And we think that's part of the reason Melissa alluded to it earlier. In certain situations, it's much easier and probably more beneficial to really do social distancing behavioral modification. In certain industries where there is no way to do behavior modification, for example, we spoke with some representatives of a migrant farm working group out here. I'm in California on the Central Coast. These individuals have to work very closely to one another. There's no social distancing available. They often live together in close quarters. In that instances, we have to figure out economical and thoughtful ways of testing them. Also, for example, the Screen Actors Guild came out with very specific guidelines about testing and are requiring actually weekly testing for most of their crew, and for their actors who are actually face-to-face, the testing cadence is much more stringent. So there are industries where this is really important. Melissa is very skilled in thinking about contact tracing and possibly pool sampling, and that's something that we talk to employers about when they think about how their employees are situated, where they're in pods or close together, where social distancing and PPE might not take care of all of the issues. And that's really what we want to help employers think through.
I will say that while I've stated that this is our last call, I'm just taking the temperature of the audience. I might, from time to time, send you an invitation for a one-off, because I think this topic is far too rich to cover in our time this morning, and it's too evolving and too important. I'm getting some enthusiastic replies, so thank you, folks.
Yeah, we agree, and that's why we're hoping your audience can help us think about this too, because as Melissa said, you know, the initial response is just to put their head in the sand and say, we're not going back to work. We hear a lot of that. We're not going to go back to work until at least Q1 of 2021. We can work remotely, whatever that looks like. But as things are starting to open up, we need to be thoughtful about how we bring the workforce back into place, and getting thought leaders like you've brought together here is really, really helpful to think about this problem.
You know, I do think that forums like this, Joe, I commend you, because if you get a bunch of people that, you know, smart people in a room, and we can come to a consensus and be a solution-driven group, I think there's nothing better we can do for society at this time than to help everyone, you know, not even for the sake of us as individuals, but for the sake of the economy, for the sake of the society as a whole. We can solve some big problems here if we put our heads together. So forums like this are so valuable. And I'm sad to hear that this is the last one, because I do think as a collective group, we have some incredible minds with incredible networks and incredible visibility into this market. And with that, you know, if we coordinate ourselves and we can get ourselves some momentum and a path towards a solution, we have a lot of power to make change here. And so as much as you can, I encourage to keep these dialogues going, and maybe even target and focus them more around what can we do as a collective to help, you know, even if it's from a political point of view or you know, an information point of view, or be models of how to bring your workforce back online. Set the model, set the tone of what's good and what's right for the community and for the employees and employers. There's nothing better than to be kind of the gold standard, and that needs to be established so that everyone else can find a path forward.
I'll say I'm getting encouraging feedback from what you're saying. A couple of things. One is everyone, including you guys, and I'll be sure to follow up with you specifically. I am keeping the 425 person strong chat room open, so we can talk all the time. It's just a different medium. But second, I'm curious to know, and there are a lot of questions piling up, but I'm curious to know, over this period of time, a lot of people, unsurprisingly, have said, Joe, can you help me get PPE? And I have come in contact with folks where I pretty much have direct communication with 3M and access to the former head of FEMA. These are very credible players with massive access to PPE. So if there's someone on the line who, I'm talking like for the state of California, for the Kaiser Health System, billions of units, or as small as thousands of units. So if you do have that need, but I bring that up in the context of there are individual manufacturers of, let's say, masks, and there are many people making masks, and they are bidding for business with California to be the exclusive mask provider for California. Is that the likely model for diagnostic tests as well? Is there some part of your organization that's right now talking to the state of Wisconsin to be the provider of tests for the state? Is it a federal thing? Is it a local? Is it a business by business? How do you do this? And from a marketing perspective, I know this is neither of where you guys focus, but from a marketing perspective, as we started at the top of the hour, I'm fascinated at how we are going to persuade individuals to believe that tests even work. In fact, Kathleen asks, what's the point of having a test if COVID-19 won't necessarily offer immunity to a new infection? I mean, that's a side question, but it's like, okay, to me, it's like, I used to work in ECGs and cardiac stress and the like, and in the beginning, I was like, why do you have both of them? And they explained to me, and everyone on the phone likely knows, when you take an ECG, that's a point in time. Cardiac stress shows how your heart works over a period of time, so you get lots of data points. If I go get a test right now and I'm fine, and I'd go to the store on the way home. By the time I get home, I could have contracted. I mean, so even the concept of testing, unless there was some kind of wearable, which is so far off, I don't even know how that would be adopted. I don't know how to. I'm not smart enough. Maybe you guys are.
Well, no, I mean, this is the whole challenge, and this even goes all the way back to biodefense. You know, I have a background. I spent many years working for the Secretary of Defense in biodefense and pandemic preparation, and it was the time, you know, after those anthrax letters, and we were putting sensors all over the tops of buildings and looking for Bacillus anthracis, but we were detecting Bacillus cereus because that's on all the soil, and it was all these false positives all the time, and it just, what it did is it overloaded the system, and nobody believed anything when it went off. Humans are the best sensors. We're going to get sick if it's out there, and we're going to be reactive. And so, I mean, that's from a bio defense perspective, but the bottom line is we have to me, being reactionary, knowing how to treat, knowing how to diagnose rapidly. I mean, you can't just get tested all the time. It's an endless state and an endless money drag. You have to be smart. You have to wear your PPE. You have to act appropriately and own the responsibility that you have as an individual to minimize the risk of infecting other people. And if everyone can do that as a community, then I think testing just becomes a secondary level, you know, not a diagnostic level of information gathering, and then it does just become a data point. But like you said, if you want to bring your workforce back, you can't. It is financially unfeasible for the foreseeable future to test 1 to 2 days a week everyone in your workforce. It just doesn't make sense. So how do you do this? How do you do it better? Well, you identify risk categories, risk groups. You arrange your population inside of your workforce in such a way where people who are at low risk of having a very significant infection, but at high risk of exposure because they're out and about all the time, you know, you separate those people from the people who are susceptible or have underlying health conditions that could become very sick from COVID. Some of those simple decisions and ways of aligning and organizing your staff can add significant differences, not only to the comfort levels of the employees and their willingness to come back to work, but also to just providing a safer environment for employees to come into. And then with regards to antibody testing, it's so frustrating because if you look back at the SARS epidemic and you go to the research that's out there, they show 3 years of immunity after SARS and MERS. You know, so the data is out there. This could be different, yes, it could. But there's a high likelihood that people who in my opinion, this is not validated in any data that I have tested, but there's a high likelihood if you've had COVID and you have antibodies, that there is an extended period of time right now during this current crisis that you may not catch it again. And it's going to be different for every person because not every person's immune system responds exactly the same. Not everyone has the same healthy level of immune response as everyone else. So you can't make these kind of blanket statements that if you've had it, you have immunity. And I get that as a scientist. But I do also think we're underplaying the effectiveness of understanding who has immunity and what we can glean from that just in a very near term, you know, just for, even if we can say for 3 months, something, but nobody will come out on the cliff and make that statement. And so I just, I wonder, like,
Kathleen follows up and she says, why should I care if I had the disease in the past if data doesn't show that I'm necessarily immune? Okay, I have antibodies.
It doesn't show that you're not.
What does that really mean? What does it mean?
It means that no one in a position of leadership is willing to take the risk to say, if you have antibodies, you have some kind of conferred immunity for a period of time of at least, let's say, based on my best guess being an expert in this field, I'm going to give it 3 months. Nobody will take that risk.
Would you?
Nobody wants to say it.
Would you? If you were the president?
Who cares what I say?
No, but
like,
here's the point.
Yeah.
I mean,
if
anyone,
I had COVID
I could be an
and I
authority
wanted to
figure
go to
and
the grocery
say,
store, yeah.
And we have authority figures who have given us questionable instruction on masks and hydroxychloroquine and other things. So,
okay, this is what I would say. If you're a healthy person,
who can you even believe at this point? What's a fact?
If you're a healthy person and you've had COVID and you've had an antibody test that indicates you have IgG at least, I mean, IgM would be early in the infection anyway. as myself, I would not be worried about getting COVID, and I would still wear my mask because the people around me in my community would look at me like I'm a murderer if I don't. But I wouldn't be concerned about in the near term catching it again. Now, I know, with strains mutate and other things can happen. Like there's no certainty around it. But I feel like the healthy population, I go back to this concept of herd immunity, and I know that some healthy people are dying, but it's like the best thing we can do is get this herd immunity, and we're going to protect the most vulnerable if those that are not vulnerable get immunity, because the virus won't be able to transmit the way that it's doing today. And initially the UK was going to take that strategy. Herd immunity was the way to go. And then they started to look at some of the unpredicted death rates that were happening with younger people, and they thought, oh, that's too big of a risk. But, sometimes we have
to... Let me redirect you just...
Yeah, go ahead.
Let me redirect you just a little bit. A question about your product. My friend Eddie asks, did I miss the discussion of your false positive, false negative percentages? And I don't think we covered that. Can you talk about that?
Yep, I can talk about that. Sure thing. So the way that I describe the false, and I'm going to speak specifically to the PCR test first. When you're so PCR, it is a fabulously sensitive assay. If you have a viral particle in the assay, it will detect it. Right now, our limit of detection is 3 copies per microliter. So you need three viral particles in a microliter in order to detect the presence of the virus and using RT-PCR. And that's been validated in our lab using multiple assays. So I'm very confident in our ability to detect the virus if it's in the sample. Now, if it's in the sample, this is where all this confusion comes from. Oh my God, PCR has all these false positives. Okay. The infection cycle and its correlation with symptoms and when symptoms show is all over the board. And so the opportunity to get a positive predictive value for a true positive is a small time window. The first couple days, it's too early. The virus probably has not made it up into your respiratory tract. And if you swab, you're getting a negative because it's not there. It's not because the assay missed it. It's because it's not in the sample yet. You haven't given enough time to kind of get up enough of it into your respiratory tract in order to detect it. On the flip side, if you go too far forward in the infection and you didn't realize you were sick, you didn't have symptoms for the first 10 days, and now all of a sudden you get a cough and you go, oh, I better get tested, and you get a negative result back. You could have had the virus for all we know. You could have been asymptomatic for 3 weeks, and you're at the very tail end. So there may be nothing in here either. If you collect between day 2 and day, let's say 10 within the cycle of infection, you're very likely to get an accurate result. It's once you get outside those windows is when the accuracy goes way down. So like after day 10, maybe you only get down to like 60% accuracy. But it's because we've had such a hard time. The symptoms are erratic. They're not always the same for everybody. They don't always indicate where a person is in the cycle of the infection. And B, it's the collection methodology and the collection time window. But the bottom line is I can say PCR, if you have the virus in there, unless there's some kind of mix up in the lab or a user error, you're going to get the answer of what was in the sample. It's very hard to miss the virus.
You need to interject. Well, you're about to ask, does that make sense? And my answer is, I'm in great pain right now. Not because of you. You're both lovely and very articulate. What my problem is what I do for a living is I help develop market strategy and communication. Basically, I'm a storyteller. I tell stories that hopefully persuade, inform, and entertain. I'm not persuaded. I'm overwhelmed. And I know you're not copywriters, and I know you're not designing a pretty, there are just too many things to communicate, to convey confidence. I'll be honest, I've been on these calls. I don't remember what IgG versus IgM is. And my friend Jim is writing me notes on the side to remind me, but I'm reasonably intelligent and I'm paying attention to this and I can't keep it in my head. So think about the most virulent anti-masker. One of the questions I threw out to my community last week was, I said, I think this is the greatest marketing challenge of our lifetime. How do you persuade an anti-masker to to wear a mask, or I don't, when you see these cavalier kids on the news saying, I don't really believe this. I think it's just overblown. It's the flu. And I'm friends privately write me, you can't fix stupid. Well, that's funny, but what about our lives here? I'm just, the only parallel I've even come close to thinking about, and I wasn't alive long enough, actually I wasn't alive at all to appreciate it, but I'm given to understand that the lobby fight against seatbelts was similar in the 60s to what we're seeing today for you can't force me to wear a mask. Well, no one seems to have a problem when you get a ticket for not wearing a seatbelt. We accept that. Oh, of course you have to wear a seatbelt. What's it going to take for, of course, you have to wear a mask? We're not going to answer it here, and I've taken us off topic, but it's just, as someone who tells stories, I'm afraid for us because
you've highlighted
boils things down to a way that everyone can understand. I don't even know where to start.
Yeah. And you've highlighted what, Melissa and I find when we're speaking to employers, a lot of it is just a dialogue and education. Hopefully, with some increased contact tracing, you have a group. I have 3 teenagers myself who, when this first came out, felt they were immune. Why do I need to wear a mask? There's been a lot of events in the news lately that people show up not wearing masks, and ideally we'll be able to trace those as a society and actually say where those contacts were when they got the disease, until we can start showing society that the 20-year-olds are susceptible, and if they're not wearing masks, they are indeed spreading it. I think that's been part of the problem. The news when it first came out was it's the elderly, it's the history of other illnesses, and the kids were saying, not my problem.
Right. Jim's writing that his 24-year-old niece has it.
Oh gosh, I'm so sorry. And that's what's happening now. So now we're starting to see the younger folks that are starting to get it, and it really is trying to take I'm just going to say this straight out, trying to take the political message out of it and really understand that this is a disease, a serious disease that we need to pay attention to, no matter what side of the fence you're on.
Let me go back to the actual product itself. Jim asks, could you talk about sensitivity, specificity, and do you have a CLIA waiver letter? Ladies?
I don't know. Yeah, I'm sure we have a CLIA. So the laboratories are the ones, that we partner with are the ones who do that. I know that they have a CLIA waiver. I'm just pulling up, my chart. I can give you the exact numbers because I have a beautiful chart here. It's just going to take me a minute to pull it out.
While you're doing that, let me ask another question. Bill asks, sure. Are you considering pooled sampling, for example, an entire classroom, test all 20 students in one test? If positive, then test individuals?
That's a great question. That's one of the things we're trying to solve, for example, for the migrant farm working population. They certainly cannot afford tests. What our farmers are doing here is trying to isolate their workers in different pods, and they're hoteling them in spaces. This would be very similar to the coal mining industry, where they've got miners that are going down into the mines for 2 weeks at a time. And Melissa and I have been talking quite extensively about how to really roll out a pooled type testing for employers so that they're testing the right sample, and that it would decrease the burden on the labs. It would decrease, the economic burden for testing, but that really is probably going to be a strategy moving forward that we'll have to address. We're finding it out here in California.
I've made you a panelist. You can share your webcam. Hi. Introduce yourself, tell us what you do, and how come your questions are so insightful.
So I'm on faculty at UCSF and part of the clinical group there. And I've been involved with some of the COVID testing, but I'm very much getting involved with sort of the going back to work problem. And so, big thing is about cost. And so that's why I asked the pooled question. So if we can start to hash or do other things to put groups together, that's going to help keep those costs down. So
you also asked about beads and bubble technology. Talk about that.
Well, you have to. It's a workflow, as Dana and Melissa know, and so the first thing you have to do is figure out the source of the material, whether it's saliva or serologic or fecal, if you want to go there. And so the first thing to do is to do that isolation or extraction, and so there are different methods to start to do that. And when you start to pool samples together, your volumes go up. So you now have to get them down to a manageable level that will go into your chip or, into your device. So the question is, about workflow. It's about thinking carefully about how you do the pooling and how you might be able to keep those costs down. And, kudos to the Seneca team. I mean, this is, these are very much needed, but it's all about the workflow. So that's why I asked the question.
Well, Bill, I want to ask you a question because we're having this discussion actually over the week. Do you consider it all prevalence in the area before you start doing pooled samples in terms of having too many positives mixed together and then you're ending up doing everything, you're rerunning everything? Or does that not really weigh in? Do we not have that level of prevalence where when you're pooling samples, let's say 3 samples at a time? And then is there a magic number in terms of prevalence that will still get you your bang for your buck when you pool?
Well, I'm taking the lessons from folks in Korea, Singapore, and other places where they've started to do this. I mentioned the fecal test because you can actually test entire neighborhoods and communities.
Yeah.
There's companies like BioBot in Boston and other places. I don't have any affiliation there, but
they're doing wastewater sampling.
You can roboticize and sort of see, I'm in San Francisco, you can see if there's an outbreak in the Castro versus the Mission Bay area or someplace else. And then the question is, okay, do you set up testing centers? How does the community respond? But that's a bigger question. On a going back to work on a company level and on a school level, testing the whole 5th grade classroom, and there's all sorts of privacy issues and other things that come into play too. But if you can sort of pool these samples in a way, maybe there's even barcoding or bead-based technologies or hydrogels that have encoded particles in them. There's all sorts of different ways we can start to think about, it's like the old Luminex technology. You can start to think about ways of, sort of breaking it down and hashing it. So you can get the 5th grade classroom, the 6th grade classroom, they might have a different kind of a barcode. And if any one of those is positive, then you got to go in and test all the individuals. But that helps hopefully winnow and lower the cost. Hope that makes sense.
It's tangential, but based on the feedback, and I'm feeling this conversation, I think what I'll do, and I'll look for folks to type in their feedback. Maybe what I'll do is, at a minimum, I'll commit to a monthly medical devices group call. It won't be exclusively coronavirus stuff. I'm sure that July's will be. But, well, I have one yay. So that's all the encouragement I need, folks. Jim asks, what kind of price point are you looking at for a test? And he gives me a range here, but I don't want to frame or anchor your answer at all. Have you already done tests?
Well, I'm happy to tell you price points, yeah.
Like what the right price point is for your product?
Right. So, volumes always matter. If we were going to come and show up on a site and do 5 samples, that's very different than coming on a site and doing 1,000 samples. So it's broken into a couple of different buckets. So first, when we do on-site collection, we bring in nurses, clinicians, and then we usually bring 1 or 2 what we call facilitators who just make sure that they set up operations and make sure things are going and every sample is accounted for. They'll do all the kind of oversight and administrative work. It just depends on how many people you have. And I think it ends up being on average, it ends up being somewhere between $20 and $40 per person to have that whole setup. And then in terms of the test, the list price for both the antibody and for the PCR test is $120. Then the list price then the bundle would be $240. But we're seeing people come in with anywhere from 800 to 2,000 samples that they want to get done. So in general, the pricing that we're able to give at volume discounts can bring it down to $99 a test. And that's kind of where we found it becomes very palatable.
$99 a test? My God, that's going to cost people a fortune. I have 1,000 employees.
Right, but what's the alternative, right? If you have a stagnant workforce that won't come back online and want some assurity that, there are industries that are willing to pay it because they have to. They don't have operations without them. And the trade-off is they don't have a business anymore. And so.
Well, fair enough, but I have to offer that you're going to be undercut. There'll be another test. And right now, as a communication person,
we'll adapt it.
How can I adequately communicate that the hematose test, I think that was the name of it, forgive me if I got it wrong.
HemaSpot.
Thank you. Versus Joe Hage's test is clinically, I mean, by the time you have clinical trials to show the efficacy versus this other test on this, because I'm ambiently aware of a test that goes for half your price point.
Yeah. Well, if someone can do it for half the price point, we can get there. I mean, when we talk about pooling samples, that's a real opportunity to reduce costs and make it far more palatable when you're looking at large groups. If there's, saliva is something that's becoming more prevalent as a sample source right now, and it's being identified in the science community as a good sample source. We are technology or assay agnostic. We want good assays that have reliable results, and that we can, put into our automation and make it as high throughput as possible to reduce costs. We're not here, we want everyone to be able to afford to do this who wants to do this. And we'll work with you, we'll figure out a way, and not only will we figure out a way to test, but a cadence and a rationale for why to test again and when to test.
Because this isn't a sales call or a promotional call, and it's a conversation. I'm letting you know that Yakov is letting me know that if you go to stopcovid.science, it's under development, but cost of materials is supposed to be $10, is what he says. And then I'm having another one.
What does that mean, the cost of materials, the cost of a test?
Cost of materials. I don't know.
Yeah, and that's what we found. That's what we found too, Joe. It's not the cost of the materials for the actual test kit are pretty reasonable. It's the assays that the labs are running right now is what's really costing. So there isn't a huge margin when you start talking about how the labs are functioning. So if the cost can come down on the lab assays to run these tests, the cost of materials, the clamshell's not expensive, a PCR vial is not expensive. Those costs are pretty reasonable. It's the other end that is causing the problems right now from a financial standpoint.
Hi, you've been really great contributor in the chat room. Nice to meet you in person. Tell us more about what you were discussing.
Oh, so these CRISPR-based tests, CRISPR being the sort of cutting-edge biotechnology, DNA editing technology, and I'm just going to put right out here that I'm speaking way out of my depths. I'm a cybersecurity guy. I'm not actually in any way an expert on biotechnology at all, but I've been following these things at sort of altruistic reasons.
As a concerned citizen.
As a concerned citizen, exactly. So it happens to be, my understanding is that these CRISPR tests won't require the PCR machines. They'll just be mixing together the chemicals and potentially have, I think there's one that's been approved already, which I think requires, multiple pots and different temperatures, and then this one, and there are a few versions of it that are being developed in different university labs. Honestly, it seems as if there just isn't enough funding and attention being put into it, unless there's something that I'm missing. I think it's just being massively underfunded, but
it's so much easier, Yakov, to go with the gold standard PCR. They know it works. This is a new technology being used in a new application, and so I think you'll feel a resistance not only from the scientific laboratory community, and it's new tools too. So people aren't necessarily equipped in the lab. It would take a laboratory transition, and then the FDA doesn't, there's, like you said, it's hard to get the funding to validate this and push it through and get all this. And I feel very bad for these really innovative technologies that have a real opportunity. Can't get there.
So we're testing and validating the Mammoth Biosciences CRISPR technology at UCSF. Charles Chu is sort of leading that effort. It uses a Cas12 instead of a Cas9. Cas9 is sort of the one that everybody sort of knows about to, edit your genome. Hopefully someday make white hair dark again, that kind of thing. But the Cas12 technology is working well. It is isothermal, and so you have different aspects of it, as Yakov points out. And what's nice is it's fast. And so, again, but it comes back to, costs, how to scale, how to roboticize, it's all about workflow.
Mm-hmm. And so, Bill, what is your sense? Is that going to hit the market in a way, and is it going to bring value and get there in time before this whole epidemic or pandemic ends? Like, can we integrate it now?
Yeah, and as you're right, the immunoassay piece is out there now. How this impacts that market and how that commercial piece. I think because of the speed, and when things start to scale, then that technology will find its market and find its place. What percentage of the total market it gets, we'll see. A lot of the proof is still in the validation, sensitivity, reproducibility, how it works in the hands of other folks that aren't experts, and so we'll see.
And what about its ability to integrate into the existing automation that the labs have for high throughput? Can it fit into the current workflows, or do you have to start from scratch?
No, it does. Again, it depends on where you are in those workflows, and how you do the sample prep, how you do. It's still the same things. RNA extraction, RNA isolation. There's still those pieces and parts, but
a couple of things just administratively here. Your host interjects. One is. If you want to share it publicly, Bill and Melissa, you clearly need to speak after this call. You can share your email addresses for all to hear, or you can find one another afterward. Second is, I think July's call needs to be a continuation about diagnostics. So we'll talk about who we need to get on that panel, because it's even broader than I think the folks assembled here.
Yeah.
Jonathan, you have.
And I just want to throw in, I would really love all these new tools that are coming online. That I'd love to do an overview and hear where everything's at and how it's emerging, because things move so fast, and people who are actually have it in their labs can give us incredible insight on how we can plan for adoption, because we'd love to do that.
Okay, I think I can help facilitate that call. Jonathan, go ahead and ask.
I have to jump off, but Joe, it's been a great discussion. Thanks for.
My pleasure.
I should brush my hair. Thanks.
Yeah, take a shower next time, Bill.
Let's get a hat.
Yeah, exactly.
Jonathan, you had a question?
Yes. Thank you all for your presentation. In following with Joe's comment about pricing, if there was a test, a PCR test or antibody test, or even an antigen test that came in at a cost of let's say $20 and had the same specificity and sensitivity that you would see in a high throughput lab, and you could get the results immediately at the place of testing, would you be shifting to that technology?
Yes.
Yeah,
New York Minute, because that is the golden solution. If you can walk away having your answers, you want to get back on a plane, you want to be able to travel, you want to be able to go stay at a hotel. If you can know instantaneously, and if the price is reasonable that you could do it first day one and day three, 2 times, $40, yeah, I would do that all the time. That would be the new way of the world. Yeah. It would almost just be like showing your ID to get into the bar, or playing a cover charge, But it has to be accurate. You have to have the sensitivity and specificity. It can't make a lot of mistakes and undermine the value of it. Early days, especially, you have to be so careful. And then if that price point is really that low, and you can turn it around in less than 30 minutes, where do I invest?
Yeah, it would be, like right now you may know this, but the state of Hawaii, in order to go on the island, you have to quarantine when you get there. You check in actually at the airport for 2 weeks in your hotel. So you can imagine what that's done to the island. What they're saying now, starting August 1st, is if you have a negative PCR test and you can, from a CLIA lab, and you go through the screening process, you can go on island and not quarantine for 2 weeks. Jonathan, your idea would solve that problem. The governor is not allowing people into the state right now who do not have a negative PCR. And if you don't have the proof, you have to sit in your hotel for 2 weeks. So what a wonderful thing to offer folks if you can do a $20 screen at the airport and get on the plane with assurances. That would be fantastic.
I invite
that is a solution that will kickstart the economy. That's what we need.
Yeah.
Can you do it? And
Yes.
where do you work?
I know Quacious friend.
Where do you work?
Phone only because he's already told me he has to go in 5 minutes. So there's going to be a limit to how many words he can cram into that period of time. Jim?
What is the clinical sensitivity and specificity of your product, ladies?
I'll make it simple. We're using the Thermo Fisher TaqMan assay.
Gotcha.
We don't have a special sauce. We didn't make up our own assay. I can give you the actual numbers if you want, but if you're familiar with that, you'll know what it is. I haven't.
I'm quasi familiar with it.
Okay, let me tell you.
So long as you have a CLIA waiver, then you have the minimum standard, FDA standard is 95 on both sides of the fence. So in order to get an EUA, an FDA 510K, or a CLIA waiver. So that's kind of where it'll evolve.
So from our clinical validation, our PPA and NPA is 100%, 30 samples though, and our specificity is 95.6. Our limit of detection is 2 copies per microliter for the TaqPath assay. We also are using a new assay called Panda. That's a pretty cool one. That's had even better sensitivity. We're seeing 0.1 copies per microliter of sensitivity there. Our specificity so far on 34, I think, samples is 100% with 100% PPA and MPA. But again, these numbers are small. It's still, great, 30 samples, but that is not reflective of what it's really going to look like at the end of the day. So that's why I hate these questions about that, because it's like it's on 30 samples.
That's the insert that goes into the box for each and every single test kit, and that's the clinical sensitivity and specificity, right? So, and they're all done on 30 samples, and that's it. Period. End of story.
That's it.
Universal.
Yeah.
Yeah,
With some of the IgM and IgG assays, you're getting larger samples. You'll see like a sample pool of like 526 where they're doing like differentiating IgG and IgM. They're doing a little more work there. Just I think it's harder. But for the PCR, it's just it's cut and dry, and they don't do more because they don't have to do more. What I want to see and the data that I look at the most is when people do large clinical studies and you have 500 people. Those are the research studies that I search out to really get an idea of how good these assays are, because what you see from the even in what we're required to do to get to the FDA as a laboratory, it's just so minuscule. It better be 100% on 30 people.
And in all honesty, I also market a serology test kit. It's a lateral flow rapid test kit, under 10 minutes results. And I don't know, Jonathan, that kind of sounds like what you're doing too.
Yes, but again, I think that there's been so much kind of bad noise in the market, poor quality, and we saw that early on. So we just kind of laid low and spent more of our time doing clinical research and good studies, and trying to understand the user component of it as well, and then just lining up the relationship so that when things, if it's really going to extend, then we'll go with confidence with the product rather than just rushing to try to meet a market demand where we don't know if we can actually serve it properly.
You both are antibody lateral flow sticks. Jonathan, you're also, so nobody has, oh man, I thought that was PCR, the $20
and PCR
20 minutes.
You're PCR, Jonathan? Yeah. I'm serology antibody.
And you know what? Right. And I am confused. And I represent consumers who don't know what the hell you're talking about. All I know is I heard that I need to get a test, and half of them are wrong. And if each of the 3 of you, my friends now, say, hey, Joe, how would you help me tell this story? And if each of you came to me, I'd be like, well, first I have a conflict, but second, if I didn't have a conflict, I don't know how I'm going to distinguish the Jim product versus the Jonathan product versus the Melissa product. In a way,
it's easy. You've got PCR, which tests your present bodies.
You've already lost me, James. You've
Okay.
already lost me. It's easy. You've got PCR. I don't know what
It's easy.
PCR
You've
means.
got the virus.
Okay, you've got a laboratory. You've got a laboratory test, one that determines whether or not the virus is present in the sample.
Melissa. It's not that I'm not retaining.
I understand what Joe's saying, and
saying
Joe,
that
it's really simple.
I'm going to, a rally.
Joe,
and-
it's really simple. Either you've got the virus on you, or we find out a little bit more about how the virus has impacted you.
Mhm.
That's pretty much what it boils down to. So you don't have the virus, you have the virus, you did have the virus.
Great, and all three of you can help me do that, right?
Right.
Yes.
Now what? You each, and it's the amount of investment going in and who's going to win and the like. Yakov, you had your hand raised a while ago. Is your question still relevant? God bless you. Thank you for waiting.
I think so. Basically, my question would be, because you mentioned that you would jump on board a $20 test. Supposing that the FDA is slow walking everything, as the economist Paul Romer has been continuously pointing out, they've been doing all along, and it's been doing tremendous damage, their inaction. Supposing, and I'd have to run this by a lawyer to see if I'm right, but supposing that just as with medical marijuana, where the federal government still has a complete ban on it, but the states say, well, we're going to allow it, and the feds don't really do anything about it. Supposing that that were true with testing as well, such that a state could, and suppose you had a state that was willing to authorize a test to be a little more bold, take the, and as I say, I'm a hacker, so take the Silicon Valley ethos, the hacker, I'm not in Silicon Valley, but take that ethos, take the hacker ethos, and try something a little bold. And I would say, in terms of the, well, what if it's not as accurate? If we're talking about screening asymptomatic people, right? People who, I mean, obviously, somebody has a cough, go get the best test you can get every time, right? If you are, right now, you think you're well, right? You're not going to pay $100. I mean, you may not be willing to pay $100. Certainly, you're probably not going to want to pay $100 twice a week to do it properly. Or if everyone would test, twice a week, we'd be done with this thing in a few weeks. Because you'd identify all the asymptomatic carriers and you'd get them out of the population, tell them to stay home. And even if you can't get all of them, let's say you get 80% of them, and that would be a pretty bad test. Or imagine a test that only caught 80% of positives. But if we're only talking about screening asymptomatic patients, you take 80% of them out of the population on a routine basis, and we could be done very, very fast. So to boil this all down to a question, would you or anybody else on the call be interested in adopting that sort of Silicon Valley ethos, moving fast, taking something that the FDA will not approve, under the guise, assuming that we could find a state that is willing to allow it and shelter under their authorization? Would any of you be willing to do that?
Yakov, that's kind of what the EUA is all about. It's this emergency use is basically like a blessing from the federal agencies saying, okay, go for it, just try to do it in a rational way, and then we'll kind of, and you can see now the FDA is tightening down to more qualitative tests.
But even to get that,
there's only 5 approved vendors in the United States for a lateral flow serology test kit. Only 5. I have one of the products. And it's even fewer for a lateral flow PCR test that Jonathan's marketing. I think, what is it, 3, Jonathan? Maybe 4?
Yeah, there's not many, and we're not officially yet on the list, but we expect to be there.
Is anyone on the call, not necessarily on the speaker, but in the audience?
Yakov, I want to answer your actual question.
Is there anyone on the call in the market for tests for their workplace? If you'd leave me a note. Melissa, sorry.
No, I just want to answer Yakov's question directly. If there was a product, let's say the EUA, we found out that they're not accepting any more EUA applications for COVID. So there was no path forward there. And you had an innovative product. I would say I would stand behind you if you had a couple very good academic institutions or medical research institutions that did some very good comprehensive studies, but like fast, it doesn't have to be, but real studies with real people in real situations. And if you had that data package and you had your core, I wouldn't believe you just as a person who owns a product, because I work in biotech. Yeah.
It is what it is.
Yeah, I know how we like to cherry pick. But if you had a couple really good research academics institutions behind you, and you were able to, I would say, let's bring it to the governor's office. Let's get it approved. If there was a path forward to do that, I would stand behind you, as long as I knew the it was real and we had confidence in its ability.
And the lawyer in me is having chest pain right now, so I am a lawyer as well.
He's a physician and a lawyer.
Complete liability.
Yeah, Jakob, you're giving me chest pain. The lawyer in me. So I mean, it would be.
He has that effect on women.
Yeah, that's a landscape that you start to hear employers already concerned about the liability that they are anticipating that they're going to have. So to Melissa's point, there would have to be a very significant path forward where an employer would be able to say, because this, I didn't go with what is FDA approved because I know this works just as well and it's cheaper. There'll be a battle there, but that's where you start to get into the nitpicking about, from a risk management standpoint, how am I going to address that topic?
But Dana, what if we didn't do it for employers? What if we took the direct-to-consumer model, genetic fitness testing, and we did it with the COVID testing, and we went directly to the consumer, not so much to inform businesses. I mean, businesses could choose to pay attention to that information or not. But I think if we did the, 23andMe model with a test like this, and it was an individual choice, there's a lot of moving parts, like where does public health weigh in? How do you report if you're doing it, if you can do it at your own home? Like, how does this information get relayed? And
I like her.
Yeah, and there's plenty of examples like that exist. The model exists, so it should be, effective to replicate.
Yeah, you just have to make sure, because there's a very different risk between 23andMe being wrong. Right. And a test for a virus that is this virulent being wrong. So,
what is really the risk? At the end of the day, you can't get tested or it's too expensive to get tested. So at least you have a data point. As long as the science is valid, as an individual, I get a data point and I can make my own choices, and hopefully I'll be smart about it. Better than having no data point. I'm sorry.
Thank you.
We could talk about this philosophically.
And tell people.
Thank you so much. Nice to meet you all.
This might be for us to wrap, because clearly we're not going
Jonathan Saul, I need to talk to you. How do I get his contact details? Okay, find me.
I know everybody.
Melissa May.
Ask me.
Joe knows everyone. He's the.
Mark Bodley writes, this is the same in South Africa at the moment.
Oh, very nice. Okay. And are you going to smack me after this? For being too on the, gonna be like, woman.
You guys are in different locations.
We'll have our risk management discussion later, Melissa.
Dana always keeps me on the straight and narrow. I need her so badly.
It's funny, as we came into this call, I was thinking, I'm tired. Boy, am I glad this is the last one. That's what I was privately thinking. And now I'm like, yeah, this isn't the last one. This is
too fun. I love dialogues like this. We got to do this more often. Pick a topic and chew it up.
Okay, so what are you doing later today? Oh, you're probably busy.
Is this a dinner proposal or?
An hour and a half long. Thank you for joining us. Jeremy popped in and out. Thank you for coordinating this. And whether you showed up on screen or not, you're part of this family. And the chat room is open all the time, so we can stay as tight as you like. My green light's always on pretty much. And this is Joe H for now. Thank you, everybody. We'll be talking more. Thanks so much for today.
Thank you.
Thank you.
Thank you.
Bye. Thanks, everybody.